Someone asked me a question about a conflict discrepancy in 2 slides, and I think that it was almost $1 million question. Where is Sarah? Is it here? All right, she's hiding, she's shy. But she asked a question. I just answered that you should be a glycobiologist asking that question. So I'm just going to go that and then start with that. I will post I just answered with a PowerPoint slide. I can post that. It is not needed, but if somebody is interested, you can look at that. So here, what we are trying to see that if you just look at the complex processing of N-linked oligosaccharide, you have these tri-mannose and you have all these different things. Now, it can be processed in the Golgi in a little bit different manner as well, high mannose, and we did not talk about it. So you can see that there are, so you only just focus onto these blue residues in the mannose. In the next slide, what we are looking at, that these are the different things, and once it goes into the Golgi, if you see that here we are looking at an example where you have this 5 mannose, which is trimmed to the tri-mannose right here. And what we talked about, that once it goes into the tri-mannose, at that point it becomes endo-H resistant, but endo-D sensitive. So this is the tri-mannose. Now remember that other than this, there are different configurations possible because you have the high mannose residues as well. So they work a little bit different. So this is now you have 3. So this is endo-D. So you only look at the endo-D sensitivity with a 3 mannose. Now if we just go to this picture, what she asked, that here you have 5 mannose and you have endoglycosidase D, why it is chopping this off? And I think that's a wonderful, wonderful observation, and she has every right to ask that, because the previous question right here is endo-D sensitive with 3, and here endo-D sensitivity with 5. So what happens is that, so the first question, there is 2 parts of the question. So the first thing is that when you have an ER to Golgi transfer, why these guys did not use endo-H? That's the first question. So they could have used endo-H, and then it will chop off the 5 mannose. Now, the problem is technical. Think about that how you are isolating this VSVG labeled protein. You are taking the cell, you are crushing it up, and then you are just allowing the sucrose density gradient after initial clarification, which will be, so let's take a look at that. So you will have all these Golgi all together, and these will again form the microsomes. Really, in your mixture, you cannot isolate separately just this cis Golgi, where it is your protein is endo-H sensitive, and you cannot isolate these separately, which is a trans Golgi, where it is endo-H resistant and endo-D sensitive. You cannot separate that because these will be all formed together in the microsome, and they do not really have any different density. So you're going to collect all of them. So you will have a mixture of endo-D sensitive and endo-DH sensitive or resistant. You have all these mixtures. The problem is that if you use the endo-H sensitive, and if you try to do this experiment in the next, right here. Oh, I'm just going back, sorry. Yeah, if you do this experiment with the endo-H sensitive, then what happens that in the 6, when you have these 5 mannose, it will digest this. It will be perfectly fine. But problem is that in the mixture, remember that the vesicles are breaking up and again coalescing, and some of these proteins, which just for this transient time moved into the Golgi, they may be entrapped into a trans Golgi where you have endo-D. So you have all these different, and also before that, remember that you will have this Golgi alpha manosidase, these enzymes. Then you have N-acetyl glucosyl transferase, alpha manosidase. So some of these enzymes will already have trimmed out into those microsome into the 3-stage 3 mannose condition, and there will be no endo-H sensitivity for them. So when you do this experiment with your endo-H, there will be a good number of these glycosylations which will not be cleaved out because they have already transformed into the 3 mannose residues. So what we do in reality to avoid this process, you collect all these residues together, and then what you do that you treat them with endo-D-glycosylase. So the question is that if you have these 5 residues, then endo-D will not be able to digest it. But the trick is when you commercial, and they actually did this experiment with this. So let me just take a little bit different. So this is 1987, remember that. So when they did this experiment, you can see that always when it has to be moved towards the medial part and the transgolgi, you still have this upper band left in here. And that's the reason that it shows that your lower band is increasing in the size. That means that it has become a little bit more of endo-D sensitivity. But you still have the ones which are not cleaved out are probably in this 5 conditions. That's where they were not cleaved out. Now, this is an experiment of 1987. When the authors wrote the book in 2016, things have completely changed. So this exactly doesn't mean this. So now how you do this experiment, I have to go to a separate slide. And I will post that. I mean, like, you really do not have to go into the detail of that. So this is the enzyme that we buy from the NEB, New England Biolabs. This is the enzyme that comes in a vial. Now, I really do not have to go into that detail. But the thing is that all you have to see is that when you get this endo-D in your tube and you do this digestion, it contains this, it contains this alpha-1,2 manosidase. See that it is already added in the reaction mixture. So what happens? That if you have some of these 5 residue mannose, it's going to chop up all these 2 mannose and it's going to get digested by the endo-D. So all of the glycosylation will come out. Everything is glycosylated. So the picture in your book, and again, I will post that, but again, not needed. But that's the reality that why these there is a discrepancy in here, because these books were written in 2016. That's the edition. And at that time, this experiment is perfectly fine because you just use that alpha manosidase 2. But here, they really should have only the thick lower band. At that time, 1987, never this alpha manosidase 1 or 2, I guess, was not added in there. That's why you still have this upper band available in there, because they are endo-D resistant. Does it make sense or doesn't? So I mean, like, we are not going to ask any questions regarding that, but that's in reality that when you work in the laboratory with these, that how things are a little bit, and there's no conflicting information. It is just the technical detail. That's why they are in there. So that's about it. Any questions about that? And again, I mean, like, it is just completely out of your interest. If you're just interested, you can just go back and read that material, and that will be perfectly fine. So let's start today's material. We are going to go to the chapter 3, and let's start with a clicker question. That will be interesting. Previous material clicker question. Let's start that. So a transmembrane protein was synthesized in the orientation shown on the left. So you can see that on the left, the N-terminal is in the cytosol, whereas the C-terminal in the ER. Now, which of the following accurately depicts the orientation of this protein as it travels to the plasma membrane? So from ER to the Golgi to the plasma membrane, how this protein is going to be facing the N or C terminal facing what? So let's see. It's not an easy question. It's a difficult question, could be a little bit, because if you have not looked into the material, but let's see. 137, do you need more time? Okay. Take your time. 3 minutes, stopping it now. There you go. Let's see. Good, but not a lot. I mean, like if you can see, 64% wisely chose C, which is the correct answer. But you can see still B is 18%. So I mean, like 64% is not a good percentage. That is that was your initial exam percentage before I threw out the questions. So I mean, like, so make sure that these are some of the questions. And if you guys, no one goes to the discussion board. I know that's boring. So I just every day go and check by myself, and then it's just like it's a lonely place. No one talks. But if you see that I posted a hypothetical protein sequence, and where you are given a chart, hydrophobic amino acid plus minus positively charged, negatively charged amino acid, and told that you just make a hypothetical protein and tell what it is. Practice that constantly with your friends. So you will be given a chart of amino acid and a sequence in the amino acid, and you have to tell it that which sequence is in where. So make sure that you do this exercise, very important, and the discussion board or study groups are very important for that. So as you can see that, again, remember that when you are talking about the cytosol is N-terminal, that is always inside the cytosol and it never changes. Cytosol is always cytosol. Whereas the ER lumen is the C-terminal terminus, that will be the lumen of the ER, that will be the lumen of the Golgi. Only at the plasma membrane, when it flips out, this ER lumen is going to change into the exoplasmic phase. That's it. So if you look here, the cytosol C-terminal, not going to work because right here, this is the ER lumen. On the cytosol right here is the C-terminal. That's not going to work either. So in the trans-Golgi network, you can see that cytosol is the N-terminal. That is the N-terminal. And trans-Golgi network right here is the C-terminal, is in the C-terminal just like in the ER. And that is the correct sequence right here. On the other hand, see that in the cytosol, it is a C-terminal and N-terminal. That also is not correct. So it may take a little while, and that's why you spend almost 5 minutes on that just single question, but these are some of the very important questions for your exam too. Let's do the second one. So the second one is you're policing the cis Golgi and encounter some proteins. You were informed that subproteins accidentally escaped the ER. What information you are going to check on the ID of these proteins to return them to the ER? This is an easy question. This is just a recap. All right, now stopping it now, 1:45. We spent a lot of time on this. Okay? Stopping it now. Good. This is 81%. I did not expect the 10 4 or 2% in there. It is the KDEL sequence. That is the ID. This is just a very simple recap question. All right, so let's start the real material. So first, what we are going to do, not a difficult class at all. So many of the things you know, just there are a couple of different things that we are going to define. And then before the class, someone asked is that how these things are actually going to happen, the mechanism of these transport of these vesicles, that we are going to talk about in the next class. There is a correction, I'll tell you that, and I uploaded the correct file. So if you just want one time overwrite the previous version of your PowerPoint, it's just instead of 2:1, but I will point it out. You do not have to do anything in here. It is not major. So let's see that what we are doing here. So first, we talked about the Golgi, what is happening, all the fun in Golgi. Now they are coming out of the Golgi from the trans phase, and the trafficking is towards the lysosome. So this is a lysosome. You have all the hydrolases. And what we mean by the hydrolases, these are responsible for breaking down pretty much everything that the cell uses for the metabolism, or we use for metabolism. Nucleases, proteases, glycosidases. So the reaction, why they're called hydrolases? Any biochemistry? Hydrolases? Why does hydrolase? Anyone? Just need one, 2 words will work. Need something. Need what? To do the breakdown by the What? Water. That's right. Water. That's the hydrolyze. So so if I hope that you have this synthesize and the degradation reactions. So when you synthesize or join two peptide bonds, one water goes out. When you break it down, you add that water. That's why these are all called hydrolyses. It's a general reaction. All of these need the water to be added to break these down. So essential for this. But another important thing is that remember that the pH inside the lysosome is 5. That is where they work. And your V-class pump that we talked about, that's a proton pump, pumps the protons from the cytosol into this. Now, why these don't digest the plasma membrane of, not the plasma membrane, the lysosomal membrane, because the lysosomal membranes are heavily glycosylated, and these help the integrity of the lysosome. The reason is that you have all the lipases, proteases, they can easily digest the membrane, but as they cannot access the membrane, they are absolutely fine. But sometimes if somehow they leak out, there is also a second safety net, as I said. The pH of the cytosol is pH 7.2. So at that pH, these don't work that well. So that's one of the advantage of that. So how these go in there, or these enzymes are targeted into the lysosomes, that's what we are going to talk about. This is a plant vacuole. It's a similar organelle, and we talked about these as well. This is a storage material, storage organelle for the plants as well. On the other hand, you have an additional thing that these vacuoles do, which is almost 90% of the cell, is to maintain the turgor pressure, to maintain the integrity and the shape of the plants as well. Similar organism. So here you can see that the lysosome does different functions. The first thing that what we have seen, we're going to talk about that ER to Golgi, and from Golgi how the things are going to be transported into the lysosome. Then we're going to talk about the two parts of endocytosis, the phagocytosis and the pinocytosis. Autophagy we are not going to consider in this course though. So let's look at the traffic, that how it is transported, the trans-Golgi vesicles into the lysosome. So again, this is very signal oriented. You have an enzyme which is known as an N-acetylglucosamine phosphotransferase. This enzyme have two different pockets. One of the pocket right here recognize a signal sequence. A signal recognition sequence. Don't use the word signal sequence because it is exclusively used for the ER targeting from the for the proteins. So there is a recognition sequence here, and these are something like this, like a DXXLL or DEXXLI. Now remember that leucine, isoleucine, very similar, so that's why these are called the dileucine motifs. Or there is a tyrosine based motif, which is Y, then XX means any amino acid, and the phi is a hydrophobic residue. So these are just not one. Remember that there are multiple of these signals, because that's the reason that you see this pink color. There are multiple signals throughout the protein, and it doesn't need to recognize every individual of these sequences. And that actually helps that if there is a mutation or something like that in here, one sequence is lost, there will be the other ones which can take care of that. So that's the advantage of having the multiple sequences, recognition sequences. So all these lysosomal trans-Golgi proteins, which are already modified with these glycans right here, they will bind to this recognition site. And then what happens right here is the catalytic site, which will get N-acetylglucosamine attached with two phosphates. And that phosphate and the N-acetylglucosamine in red, that tiny dot, is going to transfer to the mannose 6 phosphate. So the 6 carbon, it is transferred. So now this protein will be ejected out, which will have a mannose 6 phosphate residue, and that's the signal for the lysosomal targeting. So for targeting, these recognition sequences are not required. That is required only for the enzyme to put a mannose 6 phosphate in there. And that mannose 6 phosphate, or M6P, that's the targeting. So this is just another rendition of the whole thing. And again, you may have a little bit of conflicting idea here that we talked about, that there are tri-manos, and why here we have 6 manos past the trans-Golgi. So remember that we did not talk about the high manos. High manos may have entirely different type of configuration rather than N-acetylglucosamine based manoses. So again, don't worry about these extra manoses. But it is the same thing. Even if it's a high manose, you just add the N-acetylglucosamine, it's going to be targeted at the same place. So what happens now? You have the transphase of the Golgi, where you have the particular lysosomal enzyme, and these lysosomal enzymes are going to bud off into a particular region. And in this particular region, what you are seeing, that they will be associated to a particular area where you have an M6P receptor. This M6P receptor is present on the transphase of the Golgi. So on the transphase of the Golgi, these receptors are present. When these M6P labeled proteins get attached to that, it will bud off into what is called a clathrin-coated vesicle. And how this bud is formed and what is the role of clathrin and other protein, we're going to talk that entire mechanism in the next class. And it buds off. Now at this point, you have a structure, clathrin-coated vesicle, with all the different hydrolases with the M6P signal in there. It's in this coated vesicle. Now this vesicle uncoats, and this is a complete lysosome where you have this receptor still facing towards the inner side, and this particular receptor is now going to get recycled back through this late endosome. So what is the difference in between these? So right here, what you are seeing is the receptor as well as the mannose 6-phosphate related, all the hydrolases are packed into this. This goes into a condition which is the late endosome. Now, this book is not that detailed, and we will explain that. So the late endosome, at this point, think about that, there is nothing else fused to this late endosome yet. And what it means, if it may sound a little bit funny, what we mean by nothing fused to that, these endosomes actually fuse with something that is coming into the cell. But here in this late endosome, nothing fused yet. So at this point, from this late endosome, which is going to get acidic, so the only difference in between this lysosome, just out of the clathrin coat and the late endosome, is that here they're constantly being pumped this pH, and the pH is rapidly going down towards the 5. That's the important thing. And at that time, this receptor is going to get recycled back to the trans-Golgi. So no receptor, and this is a lysosome just waiting to be fused with something that is coming in. So this is one of the important part. And then what will happen, that if something comes in and fuse with this endosome, it will go into the endolysosome. It is written as just only lysosome. It is just really not just simple lysosome. It is called the endolysosome, where all the digestion will happen, and that material after digestion will go into the cytosol and traffic into specific areas where they needed to go in. So here in this step in our book, it is should have been a little bit more detailed, but because there are some steps in between these two are a little bit confusing here. We will come back to this again. On the other hand, sometimes the same way as the ER proteins sometimes escape from the ER towards Golgi, some of these proteins with the M6P is going to be leaked out of the cell, and this is called the constitutive secretion. So constitutive secretion is constantly happening, and this is not receptor mediated. So some of these hydrolases are going to escape, but it is a detrimental process. Remember that the hydrolases are really going to chew up everything, and in the extracellular space you have pH 7, not 7.2, about 0.2 less, and they will start working a little bit more, not too much, but these hydrolases will still work a little bit, and that can do a considerable amount of tissue damage. So you have a safety net for that. On the cell surface also, you have these M6P receptors. So they are going to catch any of these leaked out hydrolases, and then they are going to form a clathrin-coated pit, and form a clathrin-coated vesicle, and just the same way they are going to bring those hydrolases back into the late endosome. So this is the whole process of that, how these hydrolases ultimately targeted to the lysosome. And we'll talk about this a little bit detailed next. Now here we are looking at the lysosomal storage diseases. Why we call the lysosomal diseases as storage diseases? There are more than 40. The reason we call them storage disease, because if they are not hydrolyzed or broken down, they are going to be stored in the cell. That's why we call it stored disease. So here one of them is a Tay-Sachs disease, and if you do not have this hexosaminidase A, this is a hydrolase, you cannot you cannot produce ganglioside GM2 from GM1. That is not needed. This was written as GM1 to the version that was uploaded. So if you have not downloaded the version, you will have that is written as ganglioside GM1. So which is wrong. So basically what you do not have to go into the detail, but hexosaminidase do what it does, it converts the GM1 to GM2, and from GM2 the principal storage substance is going to be GM2. So just make sure that you remember that part. On the other hand, in the Gaucher's disease, there is another enzyme which is glucocerebrosidase. That will principal storage organ is glucocerebroside, and just make sure that you remember this as well. So these are the two which are the storage disease. For the eye cell disease, it is different. So what happens in the eye cell disease, you have a problem with this N-acetyl glucosamine phosphotransferase. So the M6P or the mannose-6 phosphate signal is not put onto for the lysosomal targeting. So none of the enzyme or the hydrolases are going to be targeted at the lysosome. And that's a very, very bad situation, that all these are going to be recycled outside the cell, and severe because of the constitutive. They are going to secrete out through the constitutive secretion, and the receptors will not be able to catch them from the exoplasmic phase to bring them back into the cell, because they don't have the mannose-6 phosphate signal or the residue. So severe tissue damage happens in the eye cell disease. The only interesting part for the eye cell disease is that in the liver, all these cells work normally, only in the liver, not in other tissues. In the liver, there is a separate pathway for lysosomal targeting other than mannose-6 phosphate. And so there is a different signal. That's the reason that the liver hydrolases go properly into the lysosome, and this pathway is still not defined. Some people suggest that there is a protein called annexin that helps in that, but still very infancy, really no straightforward idea. So the eye cell disease is more of a problem of the trafficking. Without mannose-6 phosphate, which is added by the phosphotransferase, it just gets secreted out of the cell in most tissues, cannot be targeted to lysosome except the liver. So these are some of the storage diseases. So here, this actually shows that what is the problem with the N-acetyl phosphotransferase. You can see here is also a little bit of modification. Hope you understand that. So in the previous version, I had this cross just only right here, and these two crosses were added. So you will have no mannose-6 phosphate added to the glycosylation at all. So that's why they are not going to be transported to the lysosome. So after that, we are going to look at the exocytosis and the secretion, and there are two parts. One is a constitutive pathway that we already talked about, which is completely unregulated. Vesicles bud off, and they are constantly being trafficked and secreted into the exoplasmic phase. On the other hand, there is another range which is called a regulated secretion, which is hormone, neurotransmitter, digestive enzymes. So they do need a signal to be fusing with the plasma membrane. So here is a receptor, and that binds a hormone or a neurotransmitter. When this signal comes to these vesicles, only then they are going to fuse with the plasma membrane. So not always hormone neurotransmitter. It could be just an ion, say for an example, for neurotransmitters to be released from the neuron, all you need is a calcium, and once calcium comes into the cell, that releases the neurotransmitter. That's not needed, but that's a regulated secretion. And we're going to talk about the mechanism of each one of them in the next class, though. So this is the overall process. Just shows a secretory granule getting out from the pancreatic cell, electron dense, just straightforward. This is an interesting cell. This is called a mast cell. It's from rat, but we have these. These are white blood cells. They are packed with these histamine granules, and when they release, so you will have allergic reaction. So this is just a little bit extra, not needed. On the surface of this mast cell, there is an immunoglobulin receptor called IgE type. So what it does, that IgE, it binds these allergens and trigger that response to release them. And when you take your Claritin, Zyrtec, or Benadryl, they block these receptors on the surface of these mast cells so that they cannot be released, and that's the reason that they work as anti-allergic reactions. So you can easily see that how the exocytosis is working in a stimulated mast cell. Due to a mutation in the enzyme in acetyl-glucosamine phosphotransferase, it no longer can recognize the YX5 sequence in the proteins. Which of the following statement is false? The proteases having multiple DXXLL and YXX5 are likely not going to be affected. The hexokinase enzyme, which converts glucose to glucose 6-phosphate, is going to get secreted from the cell instead of going to lysosome. The liver hydrolases are not going to be affected much as they use an alternative pathway bypassing M6P lysosomal targeting. All right, stopping it now. Make sure you got that. Stopping it now, 2 minutes. Good. But 51% says B. That is the correct answer, and I will explain why A and C. A is correct. The reason, remember that, it's a false question. So which are the ones? Let's first explain that. So first, that we talked about that the proteases having the multiple DXLL, YX5 are not likely to be affected, and that is true. Remember that I said that there are multiple of these sequences. If there is a mutation in one, the other will be recognized, and there is no problem with that. See, the liver hydrolases are not going to be affected much as they can use the alternative pathway bypassing the M6P lysosomal targeting. That is true as well. That is, we talked about that in the I-cell disease. This is what happens. On the other hand, the hexokinase enzyme, which converts glucose to glucose 6-phosphate, is going to get secreted from the cell instead of going to lysosome. This question needs a little bit of understanding of your biochemistry background. Is glucose, so the glucose, it is changing from glucose to glucose 6-phosphate. It is not a hydrolase. It is a kinase which adds a phosphate. It will, no matter what, it will not go to the lysosome. So that's where the difficulty of the question, that if you do not have that thought process from your background, that a kinase adds a phosphate, it does not hydrolyze anything. And that's why I said the reaction from glucose to glucose 6-phosphate. And that's why that is not a hydrolase, and it will not go to lysosome. And also another thing, also if you don't get that, then another thing is that you need to correlate some information that you got from previously. Remember that in the cell, when glucose enters through the GLUT1, 2, they convert them to glucose 6-phosphate. This happens in the cytosol. So the enzyme is not going to be secreted outside or exocytosis because glucose 6-phosphate does not form in the into the intracellular space outside the cell. It happens in the cell. So you have two different types of clues. And difficult question. This is not an easy question, but you can see that there are two ways you connect the dot. If you remember from your previous material that the glucose 6-phosphate this reaction is going to happen inside the cell, so that the glucose which is coming in is not leaked out. That's your number one clue, so it's not going to go outside. And the second one, if you remember, that the kinase is adding a phosphate, it is not hydrolyzing the glucose or breaking it down, so that's another way. So again, remember that these are difficult questions. This will not be a majority part of the question, but you will have see 10, 15, 20% of these questions which will be a little bit challenging. All right, so let's go to the next part, the endocytic pathway. We are going to look at the phagosome and the late endosome here. This is very similar that you have studied these from your Bio 101. So you have the phagocytosis, larger particles, whereas the pinocytosis is taking up water. And mostly when you just go back to your Bio 101, we say it's a drinking of the water of the cell. But remember that the difference is that we call it a fluid phase pinocytosis, but there's a huge difference. Pinocytosis is a constant process. It's constantly happening in the cell, where phagocytosis is triggered. So that's the basic difference in between these two. So who does this phagocytosis? So this is done by some professional phagocytes, which are known as a macrophage and neutrophils. All these cells circulate as white blood cells into the blood. I just talked about them, that the macrophage is called a monocyte inside the blood, a granulocyte. It comes out from the capillaries into the tissue space, and they become macrophage phagocytic cells. Defense and scavenging. Defense means if they find any bacteria, they will eat them up as well. And the neutrophils only attack the bacteria, they don't scavenge. Neutrophils are one of the granulocytes. The other two granulocytes, if you just remember from the white blood cells, you have a eosinophil, basophil, neutrophil. So the neutrophil is this one. The basophil and the eosinophil doesn't come out in the tissue space that much. So here, phagocytosis is a triggered event, and what is being shown here, you have a bacteria somehow invaded your tissue space. Immediately, it would bind the antibodies directed against it. Once this antibody is bound to that, these phagocytes these phagocytic cells will have receptors which recognize the Fc portion of the antibody. You know that very well now. So that this is the Fc region. Remember that we used to attach the fluorophores to this. And once this receptor recognizes the Fc region, along with the antibody, it will engulf the bacteria. This is the picture that a neutrophil is engulfing two bacteria that we have seen using the pseudopodia. Straightforward. We have seen this picture where the macrophage is recycling some red blood cells. You can see the pseudopods being formed. That's it. So after that, we are looking at the fluid phase endocytosis, and macrophage can take about 25% of its entire volume per hour, whereas you can see that this is really important for the lower group of particularly the protozoans. Dictyostelium is an amoeba, and antamoeba can take 75% of their entire volume in fluid per hour, because this is the process of feeding. They are getting the nutrients along with the water. So then the water is going to go out, of course, but the nutrients will be used up in the cell. So very important. So how this is formed? So there is a particular region where you have this clathrin-coated pits. Clathrin is a protein, and we are going to talk about this entirely in the next class, that what's the mechanism for that. And these clathrin-coated pits are going to get pinched off from the plasma membrane and go inside the cell. And this is the typical way that the pinocytic vesicles are formed. So here, on the other hand, is a receptor-mediated endocytosis, which is a form of pinocytosis. This will be very different from what we learned in Bio 101, because in the Bio 101 we talked about that, okay, all right, there is a receptor-mediated endocytosis, there is pinocytosis, and phagocytosis. Now you know that the phagocytosis is different because it is triggered by a signal. You have to have a receptor bound to that, which we didn't talk about that. A receptor needed to be bound to this organisms or structure, which will cause the phagocytosis. Whereas this general receptor-mediated endocytosis happens spontaneously. So these are some of the receptors, Y-shaped structures. These are not antibodies, remember that. These are not antibodies, receptors. And these receptors are scattered onto the membrane, and they are constantly taken in. They are coming into the cell constantly. And this is spontaneously happen, and they are going to get recycled back as well, but this happens all the time. Now, some of these particular macromolecule, which does have a ligand, which binds to the receptor, by chance, if it binds, it will be endocytosed as well by this process. Now, remember that whether this binds to these receptors or not, it is going to take in anyway. So this is, again, remember that your phagocytosis was triggered when a particular ligand bound to its receptor. Here, no matter what, if you have the receptors even empty, there is no ligand bound, then it's going to be phagocytosed as well. So, not phagocytosed, it's taken to the fluid phase endocytosis by this way as well. So, even if this was empty, they will go in. And this is a chance factor that if you have a ligand being bound to a clathrin-coated pit, and if it just happens, then it will be taken in as well. So, it's a chance factor and spontaneous. Classic example, this is an LDL structure, whereas you have this cholesterol inside in the esterified form, and you have the just phospholipid monolayer, which helps it to circulate through the blood, and is added with this ApoB protein, which binds to the receptor. So just a different rendition. So you have this cholesterol esters right here, which is inside, which is a dense esterified cholesterol. And then you have the phospholipid, and you have this apoprotein B in a structure. Classic pictures of real-time, not real-time, or reality in a cryo-electron microscopy. Think about this, that this is taken at physiological temperature 37 degrees Celsius. So what happens that you take these at 37 degree and you plunge it to liquid nitrogen, and it forms the vitreous ice, and it is frozen in time with 37, because if when you just take this in water and dip it in liquid nitrogen minus 296, there is no time that there is a change in the shape of the structure, so you can exactly see how they look like in there. And they are the ApoB attached onto the central core. So here what we are looking at is the uptake of the LDL, and here you can see the LDL is bound to the receptor, clathrin-coated pit form. It is going into an early endosome, then it is going to a late endosome. From the late endosome, you can see that it is recycled back to the cell surface. On the other hand, in the late endosome, the lysosomal, it's going to form the endolysosome, which is not shown in this one, and it's going to degrade all these, and you get the cholesterol, you get the fatty acid, you get the amino acids, which will be helped the cell to build the membrane. It will help the cell to use them for protein synthesis as well. This is very straightforward, that how these LDL is getting into the cell for the synthesis, and the receptor is recycled back. So this is just another experiment where you add this this iron to the LDL particles so that they become electron dense. You can artificially do that, and that just shows that right here that these are electron dense. These dots, you can see that they're electron dense, just a particular cell. Genetic disorder, there are two types. You have LDL receptor missing all throughout. On the other hand, you can have another one where you have the LDL receptors, but they cannot associate with the clathrin-coated pit. The outcome is the same. These people cannot take out the cholesterol and will have very early cardiac disease and very, very detrimental condition in these conditions. So let's take a look at the next question. This is a trick question as well. The core of the LDL particle contains fatty acid linked to the cholesterol. True or false? All right, 1 minute, stopping it now. So 64% true, 33% false. True is the correct answer. Why it is so? Who can tell that why you answered true? Because I said it's a true question? I mean like a trick question, that's why? So who gives me the answer? Anyone? Can anyone tell that what was the reason? I hope that they know. I will going to talk about that in a second. So here, look at this very carefully. This is a biochemistry prereq trick question. It's not an easy question, and I'm not going to ask directly unless I just talk about this in class like this. So here you have cholesterol esters. What are esters? You have a clue right here. You can see that this is unesterified cholesterol like this, but here you have a cholesterol ester with a tail. So what is a cholesterol ester? It is added with a fatty acid. That's a cholesterol ester. So it's going to bring the fatty acid and cholesterol both for membrane building into the cell. And again, this was not told to you. And if I don't say that like this, I will not ask this question. But that's, and you can see right here, this will not give you the clue. It shows the cholesterol and the fatty acid, but you may think that these fatty acids were interspersed with the phospholipid, and the phospholipid contains the fatty acid. So this picture will not give you the clue that until and unless you know that the ester is basically attached with the fatty acid. All right. So that was not an easy question. So now we're looking at the fate of the cell surface receptor in the endocytosis, and there are three things that can happen. So the first thing what happens is these receptors come to the early endosomes, and then they are recycled back onto the cell surface. The second they have a transcytosis, where they can go into a different area. This is a basolateral membrane. They are coming from the area of apical membrane, which is one domain, separated with the basolateral area from the tight junction, and they can just go into a different area. And they can go for degradation into the lysosome. So here, what we are looking at, we have a little bit of conflict. That's what I put this slide. This is not in your slide. So just again, not needed for the exam either, and just explaining. See that here, what happens, we talked about this receptor being recycled, LDL receptor. This is late endosome, and from the late endosome, it is being recycled back to the cell surface. Here it says early endosome. This is the Albert's molecular cell biology. Molecular cell biology is this one. This is the molecular biology of the cell from Bruce Alberts, and it shows the early endosome. So this is what is confusing, and sometimes you have this conflicting information, but both are correct. So here is a paper. This is a 2018 paper, and it's a Nature review paper, molecular cell biology, to degrade or not degrade. So see that in reality, what is happening, which is the in-between lines in reality, you have this early endosome. From the early endosome, the receptors are recycled back. This is called the fast recycling. On the other hand, you have a late endosome. From here, they are recycled back as well, which is called slow recycling. So it happens everywhere, but the authors choose just one or the other, and no one is correct, no one is wrong in that sense. But what happens, our textbook doesn't go into the detail in between lines of the information. That's why sometimes you may have this conflicting information. So if you find those, just always ask. Remember that as soon as this late endosome, which our book doesn't say, you form the late endosome, and here still at this point, the pH is going to drop, and the lysosome is going to fuse with the late endosome to form an endolysosome. If the lysosome fuses with the endolysosome and the receptor is still there, the receptor cannot survive. The receptor is going to get digested by that, because it's a transmembrane protein, and the receptor does have a C-terminal end. It's going to get recycled. It's going to get degraded into the endolysosome. So the receptor must escape the late endosome before the lysosome fuses with the late endosome. But unfortunately, what happens when you see this picture right here, this doesn't give you that detailed information. So these are some of the things, and we will not ask these conflicting questions either. So don't worry about these. So same domain recycling, we already talked about that. It is going back to the, no matter what, whether it's an early or late endosome, the receptor is taken in, going back to the same domain. Here is a very interesting thing, that it is a transferrin receptor, and see that what happens with this receptor. This receptor brings iron into every cell. Very, very important receptor. So this receptor have two configurations. One is known as an apotransferrin. It binds at the acidic pH with the receptor very, very tightly. And then there is another which is called the ferrotransferrin, which is added with the iron, and it binds with the receptor at neutral pH. So here you have the transferrin receptor on the plasma membrane on a coated pit. Here you have a neutral pH. This is outside the cell. Say suppose this is the intestinal lumen where you uptake the iron from. These transferrin molecules are free. These are moving around. These are secreted, and these are moving around into your intestine. And they catch these iron that is being moving through the intestine from your dietary iron. They catch this iron from the intestine, and they have become high affinity for these receptors at neutral pH, which is a neutral pH. So they bind to the receptor. You have a receptor-mediated endocytosis, and they end up in the late endosome. So what happens in the late endosome? You have a low pH of 5. Right here now, at the low pH, the ferrotransferrin converts into the apotransferrin, and the apotransferrin binds to the receptor very tightly, but it removes the iron which is used in the cell. So this tightly bound apotransferrin is now recycled back to the cell surface. So at acidic pH, remember that the acidic pH, it is bound at very strongly with that, recycle it back. As soon as this receptor and the transferrin complex reaches the cell surface, at that time, it's going to come off the receptor because it is now encountering neutral pH. It is tightly bound to the receptor, the apotransferrin bind to the acidic pH, but at the neutral pH, the transferrin is going to come out. So this transferrin is going to go out in the intestine once again to catch iron. As soon as it catches iron, it will be transformed into the ferrotransferrin, and that will bind to the receptor at neutral pH, so they can be taken in. So it's a very smart way that how you release a molecule into the intestine to catch iron and bring iron back into the cell. So a very classic example of receptor recycling to the same domain. On the other hand, here is a specific example. Epidermal growth factor is a signal. So this is taken up by the receptor, and the receptor sends a signal for growth inside the cell. And at that time, the signal as well as the receptor directly goes to the endolysosome, and it is completely degraded. The receptor as well as the ligand both are degraded. So this is another example of that, because after the signal is passed, you do not need that signal anymore from that, so it will be recycled. Here is another, the last one is called the transcytosis, and this happens also in particularly newborn mammals across the intestinal epithelia. So what you are seeing is a cross section of the intestinal epithelia. Here you have the intestine, lumen, pH 6. On this side, you have a blood and the interstitial fluid, pH 7, neutral pH. So here at this point, along with the mother's milk, that for the mammals, the mother's antibodies come into the intestinal lumen. And what happens in there, these antibodies are heavily glycosylated, and they are not digested by the digestive enzymes of the newborn. They are taken up by the FC portion and again into the clathrin-coated pit, and they are recycled to the other end. The difference is that at pH 6, these have high affinity for the FC region, so that's the reason that these receptors are binding to the antibody. On the other hand, at pH 7, these receptors have a low affinity for the antibody, so these antibodies are released into the blood, and these antibodies which are coming from the mother to the newborn is going to get circulated first initial part, and this gives the initial immunity from the mother's milk to the newborn. And this is known as a transcytosis, where from the apical membrane side, they are transferred to the basolateral membrane to go into the blood. All right, so here is our last clicker question. In an individual, the transferrin receptor can change its conformation from apotransferrin at neutral pH. Which of the following do you think is the most logical consequence of the mutation in that individual? The person will be able to uptake iron but will not be delivered to the cells of the body. The intestinal epithelial cells will accumulate iron in the endosomes. The person will not be able to uptake iron from the intestine. All of the above other is correct. Good, everyone? Stopping it now. Distribution is really difficult. So C, 48%, that is the correct answer. The person will not be able to uptake the iron from the intestine. And C, just give me 1 second before I'll let you guys, still you're going out 5 minutes early. So what is happening, just make sure that you, the logic, you just saw this just in the previous slide. And if you're just seeing it for the first time, I don't expect that you will completely retain that information or absorb that information yet. So I completely understand that. So remember that apotransferrin at neutral pH will come out of this. So the important part is that the transferrin receptor can change its conformation from apotransferrin at neutral pH. So the apotransferrin needs to go out from the receptor at neutral pH so that it can catch the iron, becomes ferrotransferrin. At that time, it will have a high affinity at the neutral pH. It will bind to the receptor, and it will be recycled back. So that release of the apotransferrin is not going to happen on the cell surface, so it will not be able to catch any iron. So the person able to uptake iron but will not be delivered to the cells of the body, that is not true because it will not be able to catch even the iron. Intestinal epithelial cells will accumulate iron in the endosome. That's not correct because this has nothing to do with the pH because you are not getting the iron at all. The person will not be able to uptake iron from the intestine is correct because after being recycled, so let's go back here just for a second. So after, see that here, the apotransferrin is bound tightly. The problem that I asked is that here, when the receptor goes to the cell surface at neutral pH, the receptor cannot change its conformation, so it is still tightly bound to this apotransferrin. It cannot come out from there. So if it cannot come out, it will not be able to catch iron and transform into the ferrotransferrin and cannot be taken back. So make sure that these are some of the questions which are a little bit of critical thinking. So think about these ways. So I just uploaded the video so late. That's why I thought that I will not go into the next part and we'll finish that on Tuesday. So no work yet.